Good morning, everyone. I'm going to ask my distinguished panelists to come up today to join me on the stage. Dr. Merrick, Dr. Hirsch, Dr. Baracchio, and Dr. Kirshner. Please come join me. While they're coming up to the stage, I'll say a few words to introduce this distinguished panel today. We also will have Dr. Balovanov from EMA joining remotely. I'm so honored to serve as moderator of a session that truly communicates regulatory innovation and impact in unprecedented ways. We started off today hearing from distinguished leaders at the agency, both agencies. And in the session we're going to be covering today, please note, we all know that neurodegenerative diseases truly represent a devastating public health crisis with escalating global burden that is truly unsustainable. There's an incredibly high rate of failure of therapies for clinical trials for neurodegenerative diseases as compared to other disease areas. Chronic progressive disorders, including Alzheimer's and Parkinson's disease, impact not only the patients, but the entire family and ecosystem who cares for these people, and the impact lasts a lifetime even beyond the time in which the person passes. This session has meaning to me that it's hard to express in words, as these disorders have affected my life, both personally and professionally. In my career as a drug developer, I'm convinced that my 25 years in the lab of more than 45 promising therapeutic targets for degenerative diseases could have had those treatments reach patients if such therapies could have been administered early in the disease course. Yet today, I'm excited because hope is lighting the way. The past 2 to 3 years have witnessed transformations in therapeutic development for neurodegenerative diseases in revolutionary ways that you're going to hear about in this session. The world is reconceptualizing what neurodegenerative diseases truly mean. This is due to novel biomarkers, such as imaging, biofluid, as well as genetics, that are emerged as powerful tools that illuminate in living individuals molecular pathways and signatures that can even be detected years prior to onset of clinical symptoms. Such tools allow us to have a clear roadmap to enabling advancement of precision-based medicines in these disorders that are so heterogeneous. These discoveries now illuminate a light to a pathway for early intervention. It takes bold, brave change agents to lead during times of transformation. Groundbreaking pace is being led by these esteemed leaders who join me today on this panel. To start off this session, I want to start with an individual who's had an impact on my life, personally and professionally, Dr. John Seibyl, who I've known for over 25 years. His words will resonate with us all, hopefully not only for this session, but as we lead this 3-day meeting this week. And to kick off this session, his words will capture the true essence of patient-focused drug development. So I'm going to have everyone listen to Dr. Seibyl's words today.
Good morning. My name is Dr. John Seibyl, and I'm here as a video presence to set a metaphorical table for this session to provide a context and an overview of where we are and where we might go in therapeutic trials in PD at this truly unique moment in the 200-year-plus history of research. I admit that I'm not able to be with you in person, for I'd very much like to celebrate, ruminate, and chew on options and opportunities to move forward. I do have some skin in the game as a Parkinson's disease researcher, but also as a person with Parkinson's disease. The latter has informed the former in no uncertain way, and it's been both a curse and, strangely, a blessing. When I was a boy in the early 1970s, There was a swath of posters and coffee mugs, t-shirts, and bumper stickers bearing the quote, "Today is the first day of the rest of your life." I remember thinking about the different meanings and interpretations this could have as I exercised my newly myelinating frontal cortex. Later, considering PD, that phrase might be rewritten. Today is the best day of the rest of your life. A strange bit of optimistic pessimism which speaks to the fact that the inexorable progression of this disease makes each day potentially as good as it gets. In other words, it's all downhill from here. Yet, hope does spring eternal. And two things have happened in the PD world, reflecting significant advances in research. First, we just have a better understanding of what's wrong in the brain with individuals with Parkinson's disease. That is, the accumulation of aberrant alpha-synuclein deposits in Lewy bodies within neurons. Second, we have an emerging toolbox of ways to interrogate this primary pathophysiologic event using biomarkers, which are sensitive to the onset and the elaboration of this pathology years before symptoms become manifest. In PD diagnosis, it has classically been based on identifying patterns of emerging and progressing clusters of signs and symptoms with a few imaging, laboratory, or genetic tests to rule out some diagnosis, but nothing to rule in a diagnosis. Armed now with a better understanding and a biomarker toolbox, we can improve upon this. There is hope for improved accuracy and timeliness of clinical diagnosis and serial monitoring of the clinical course. This is the bread and butter of clinical medicine, conferring accuracy in treatment planning, offering a sense of reasonable expectation of disease progression, and giving clinicians, caregivers, and patients a compass for meeting the changing medical and psychosocial needs. The significance of all this has not been lost on the clinical trials world in both Alzheimer's and Parkinson's, where the strategy is now to find people with the biomarker evidence of pathologic change, pre-manifest symptoms, and get them into a potentially disease-modifying therapeutic trial. This allows for the wild but no longer impossible speculation about the oxymoronic phrase, preventing PD, a pink champagne and caviar moment, indeed. The development of biomarkers which provide relevant and useful information encourages the development of new algorithms to approach the diagnosis and classification in the context of clinical trials. This sets the stage, or the table if you like, for the so-called biologic staging algorithms, one notable one being the NSD-ISS approach, where you take a biological definition of the disease as the new diagnosis and combine it with a clinically driven, empirical-based series of functional and or other milestones which reflect the amount of functional impairment the patient has. This is a tremendous advance in the research trials world and perhaps in the future in the clinical world. Why? Because diagnosis rooted in pathophysiology gives a stable target, more homogeneous cohorts, and more reliable and statistically valid outcome measures of efficacy, or even small numbers of misenrolled patients can affect the validity of the outcome. This provides more clarity for clinicians, drug developers, and regulators, and supports the interpretation and the integration of findings across different studies. So that's okay for regulators and clinical trialists and drug development folks. However, what about patients and their families? And what about in the future? Putting on my patient hat, I wrote for a recent publication what it was like having PD. Quite frankly, it's a quiet nightmare that doesn't end. You start each day thinking about the physical challenges in front of you and whether some new incompetence will emerge and how will one deal with it. Yet, if you can have nightmares, you can also have dreams. And I dream of a treatment that eliminates the aberrant alpha-synuclein from my brain and stops its toxic spread. Of finding a way to prevent the onset of motor symptoms in those coming along behind me. I dream that the remarkable and underappreciated caregivers receive better, more accurate prognostic information and expectations about their charges, changing functional capabilities that enlightens and supports their care. These are more possible with a biologic understanding of the disease and an empirically based clinical staging of Parkinson's disease. So I leave you with this table now set. I hope I got the salad on the correct side. And I'll also leave you a mindset of hope, gratitude, and urgency for the work ahead. In pursuit of this work, another iteration of our quote. May today be the first day of the many best days of the rest of your life. Thanks for your attention and your work, and bon appetit.
I couldn't think of a better way to start this session. And so I would love to call on each of our panelists to say a little bit about who you are. Each of you represents a unique ecosystem. And I'll start with Dr. Merrick first, who I've partnered with Dr. Seibyl for many years. Please say a little bit about your current role and what is your view right now on biologic staging and your area.
So thank you very much, Diane. And it's a pleasure to be here. Of course, it's my current role is that I continue to lead the Parkinson Progression Marker Initiative study, which is a large multi-center observational study, really sort of sponsored primarily by the Michael Fox Foundation with lots of other industry support and CPATH support, that really the goal of which is to develop biomarkers that could inform the progression of disease. It's, of course, a little bit bittersweet to see John on this video. So as always, I mean, I've for those of you who aren't aware, I've had a close partnership with John for more than 30 years, having published our first paper together in 1993 on beta-CIT, an imaging agent for Parkinson's disease, which of course ultimately led to the action of CPATH as well. And so I think that, as always, John is focused on how the science can lead us to better therapies. And that has, of course, been his focus for 30 years. Of course, the urgency of that has been greatly accelerated by his own situation, his own disease. For those of you who aren't aware, he continues to be very active in the scientific endeavor, working, as recently as last week, having had discussions, and he's writing a paper about a different imaging agent. So he continues to fight the fight, as it were, as we all should. He brings up, of course, the new work that has really generated a lot of interest and really transformed our thinking in the area of Parkinson's disease. Here again, we heard Klaus already mentioned, and Klaus and Diane should have been instrumental in the developing letter of support for alpha-synuclein as a tool to help us to develop new therapies in Parkinson's disease. This has really been another remarkable achievement by CPATH, and done in a remarkable time frame, which is, I think, speaks to the level of collaboration in the field between all of the groups involved: the Fox Foundation, CPATH, the FDA, and independent scientists have really brought this forward in a remarkably rapid fashion, which is great, because I think, as we just heard, time is not neutral for people who have these disorders. And certainly just for the development of the alpha-synuclein seeding assay as a tool has really enabled us to transform our thinking in Parkinson's disease, to develop this entity which we call neuronal synuclein disease, which really now encompasses all of synucleinopathies, and to go one step further and to develop a staging system that really encompasses both biology and clinical features. And really the goal of that really proposed staging system, which really at this point is a research system. It's a hypothesis that needs to be proven with data, which happily we can collect in the PPMI study. But the goal of that is really to accelerate therapies, both in individuals prior to the onset of symptoms, which for the first time we can actually identify and categorize and bring forward as a potential clinical trial, as well as reducing the severe problem with heterogeneity in individuals who are typically part of clinical trials. So I think we're very hopeful that this will again be a huge advance for us in enabling us to accelerate therapies.
Thank you so much, Ken. Great. Dr. Hirsch, you're leading one of the leading disease-modifying therapies recently approved for Alzheimer's. Can you comment on where you see this field today?
Sure. Did you want me to do any introducing?
Please start with yourself.
as I do that. So,
Yes.
Again, thanks for having me. I'm Steve Hirsch, as you said. I'm a vice president at ASI, where I lead the clinical development of the lecanemab program. My past before that has been as a practicing neurologist, where I've cared for patients with neurodegenerative diseases up until recently, much of that being in Huntington's disease, actually. Along with that, as part of my academic career, I've performed laboratory research, biomarker research, and participated in a lot of clinical development activity in neurodegeneration. It's pretty actually amazing to be part of this program with this remarkable therapeutic that is one of the first to demonstrate both the disease-modifying benefits and clinical benefits in one of these intractable diseases. In terms of the biological underpinnings of it, I think it's worth mentioning a few things. So first, I think without the biological definition of Alzheimer's disease, it would have been hard to make the progress that we've seen in recent years in Alzheimer's disease. I think this goes back to the FDA signaling willingness to consider biomarkers as important, and the evolution since then of taking some of those biomarkers towards surrogacy. But in terms of the development of our product, one of the really pivotal things that made it possible was being able to use a biomarker, in this case a biomarker for amyloid, to be sure that the patients that were included in the clinical trials all had Alzheimer's disease, whereas before those trials all had mixtures of patients, and that greatly diluted the power to demonstrate benefits. And then past that, those biomarkers have been really key in understanding the patient populations and understanding how benefits accrue to different subpopulations. One of the really important things I think that have come out of these studies is the capacity to use biomarkers to answer kind of the question we've always, or to confirm the thought that we've always had about these diseases, which is that earlier treatment is better. And so we've been able to confirm that in the context of our development program by showing, with the help of biomarkers, that the benefits are likely to accrue, to be stronger the earlier there's a treatment. So we've assumed that, but now we have some confirmation for that. So we also have a program in preclinical Alzheimer's disease, which are in testing therapeutics in patients who do not yet have symptoms. And being able to use biomarkers to classify them, to enroll them, to develop a screening funnel so that the patients going into those studies are the right patients has also been very critical and instrumental. One thing I would add also in terms of biomarkers is now that we have a product on the market, we're facing and learning about what it means to prescribers to have those biomarkers be mentioned in the label. It's now the drug is indicated for patients that have a biomarker, that have confirmed amyloid accumulation in the brain. There's biomarker data in the label that provides some of the information about the biological effectiveness of the treatment. That is also something that physicians and healthcare providers need to understand and need to have conversations about their patients with. And we're seeing how prescribers are reacting to both all this information for the challenges of communicating, the challenges of bringing biomarkers into shared decision making. That's been very interesting and maybe leading the way to future conversations like that we'll see in some of these other diseases.
Excellent comments. Thank you, Dr. Hirsch. Dr. Kirshner, we're eager to hear from you in your leadership role across diseases at Roche.
Sure. So I'm a neurologist as well. Before I moved to industry, I was taking care of patients in a memory clinic at Stanford. And kind of during my training and during my early days working in a memory clinic, amyloid imaging and cerebral spinal fluid markers for amyloid were first becoming available and first starting to be used in the clinic. And I remember, just to amplify, Steve, a point that you made, you know, having been fellowship trained, working at universities, it was very humbling to discover that 20 to 30% of the patients who we were very sure had Alzheimer's disease actually didn't. They didn't have any of the requisite underlying biological process going on in their brain. And I remember that being, you know, such a wake-up call to the importance of using biomarkers to understand the patients that we're treating. My role right now, so I moved to industry because I got tired of telling my patients, I know exactly what's wrong with you, but I don't have a whole lot that's going to be meaningful. And so I moved to industry to try to change that. My role right now in Roche, I'm a vice president and I lead an early development group that focuses on phase 1 and phase 2 trials of disease-modifying drugs across a wide range of neurological indications: Alzheimer's disease, Parkinson's disease, Huntington's disease, multiple sclerosis, autism, and a handful of other illnesses. And I would say that in basically in the work that we do every day, biological staging is critical because we're developing drugs that are interfacing with the underlying biological processes of the diseases that we're trying to treat. And so being able to first identify the patients who have the relevant biological markers and being able to track what it is exactly that our drugs do to those markers are essential to us being able to translate the science out of the preclinic into human populations. And as Steve also alluded to, as we understand, right, that by the time a patient has clinical manifestations, symptoms of their underlying disease, it's already quite late. The biological processes are already deeply embedded by that point. And so being able to treat earlier is essential. And in fact, being able to treat before there's outward manifestations of cognitive loss in Alzheimer's disease, motor and non-motor dysfunction in Parkinson's disease, and cognitive and motor manifestations of Huntington's disease. Being able to intervene before those things happen is essential in having biological definitions of the disease and tools that we can use first in research and eventually then also in the clinic to be able to understand what we're doing is essential to the everyday work that we do in drug development and trying to bring forward new therapies that hopefully do make a difference.
Outstanding. Thank you so much. Dr. Baracchio, please tell everyone around your background and how you view this current pioneering stage in drug development.
So I'm Teresa Baracchio, and I'm the director of the Office of Neuroscience in the Center for Drugs at FDA. And I'm also a neurologist. I did fellowship training in geriatric neurology, so I also shared many of the experiences that Dr. Kirchner described of being in clinics when biomarkers were first starting to be used in research. And also, I remember when the original bapineuzumab trials were going on, where biomarkers were not being used to define those populations, and the great disappointment in those early, well, quite a few failures of early Alzheimer's disease trials. And also at the time when there were attempts to move as early as possible in the disease and some of the earliest discussions of preventive trials in Alzheimer's disease. So I have that background that I've come to with the FDA, and then at the FDA I'm now in a position where we're giving advice to companies on how to do these trials in these populations. And so the use of biologic staging has really been integral to our advice that we're giving to companies, and has been for over a decade now. We know that drug development is this space where there's an intersection of clinical research and clinical practice. And so we are right at that intersection at the FDA. And so this information is, the use of biomarkers is critical to us as we are giving advice on how to both design the trials that would then support use in these early stage populations, and then also giving advice to practitioners and prescribers for how to use these drugs once they are approved in these populations. So having well-defined biologic-based definitions of diseases are really critical to our work. And as Dr. Kirshner said, they're really essential if you're going to move earlier into early stages of diseases, because you want to intervene, especially in neurodegenerative diseases, any progressive disease really, but we do, of course, are focused on neurodegenerative diseases up here today, want to go as early as possible in the biologic process before symptoms start, because you know that by the time the symptoms have started, a substantial decline and loss of neurons has already happened at that point. So we faced this early on with Alzheimer's disease, as those were some of the first preventive trials that we've seen in neurodegenerative diseases, and we struggled with how to define populations that are appropriate for enrollment. Some of the earlier trials did have patients who had very, very slight symptoms, so that you were able to make a diagnosis of mild cognitive impairment, perhaps, but we really would want to go earlier, before even a stage of mild cognitive impairment, and then you're using terminology like preclinical, prodromal, pre-symptomatic. It's really quite imprecise. So we've developed in our Alzheimer's disease guidance that came out in 2017, 2018, I can't quite remember the year right now, but we proposed some staging criteria in those documents for how you would define the earliest stages of the disease, that sort of moved away from such labels as preclinical and prodromal, and used 1, 2, 3, which is a more precise way of defining the population. I think that has really helped us in giving good advice to sponsors in those stages of disease. We are now, of course, facing similar issues in Parkinson's disease right now, and dementia with Lewy bodies, and the neuronal synuclein diseases, where we have a number of sponsors that are coming to us saying they want to treat early Parkinson's disease, and we're saying, well, what does that mean exactly? What do you mean? And so the advent of the NSDISS criteria, which Ken has been instrumental in putting forward, is helping us to now advance those conversations as well, and with developers as to, what is the appropriate population to enroll. I'll stop there. I think that gives a good setting the stage as to what we're dealing with right now.
Certainly, each one of you could have an entire session. Yeah. So, Dr. Balabanov, so that's why this is so rewarding for me. And Dr. Balabanov, we'd love to hear more from your background and the work that you've done pioneering with CPATH in nervous system disorders.
Thank you very much. Hello, everyone, and thank you for inviting me to this distinguished panel. My name is Pavel Balabanov. I'm also a neurologist who has more than 15 years ago moved to the regulatory side. And I now lead, for the last 6 years, I lead the office at the EMA responsible for life cycle management of drugs in the areas of psychiatry, neurology, and ophthalmology. I can only agree with what has been said by the panelists before me, and I'll just try to highlight why I am here today. And this is to reiterate the need for the regulators to be part of the discussion as early as possible and as frequent as possible. We realize that this is a period where, due to the scientific and technological complexity and the advances in the field, there will be many potential routes to be taken. And we would like to be, specifically from the regulatory side, part of the discussion which route may lead to where. This is a time for challenges, but also for opportunities, and we would like to be able to allow the right resources to be directed in the right, as much as we know, of course, in the right directions, so we can all bring as soon as possible some benefits to patients. So today, maybe we can try to address also what practically we can do in the regulatory world to support the different initiatives. But I think that at least in the area of standardization, validation, determination, the context of use of many of those proposed tools, also solving some of the ethical, practical, and other challenges in the field about accessibility of those tools and how to treat them. And last but not least, and I'm very happy to be part, as I said, of this distinguished panel, trying to address the problems that we face globally. It all begins with us being present in a dialogue of different format, present in such panels in an individual approach by researchers and academia and industry. And I just wanted to reiterate our openness to such dialogues, and I'm happy to be here once again and ready to take questions. Thank you.
Excellent, thank you so much. So I want to stay with this theme of regulatory science for a few moments. We're going to a little bit go backwards in order. So I started this session with a comment that I don't think very many stakeholders appreciate the role of regulatory agencies and champions such as we have on this panel today in driving change. And I want to start with you first, Dr. Baruch, to say a few words in the area of Alzheimer's, where biologic staging was first, to this runway. You made these comments at the Alzheimer's conference in this similar session. How do you see this juncture between drug development, research, and clinical practice when you're dealing with somewhat complex biomarkers such as this? What does that landscape look like at that intersection?
Right, so think for a second. So in Alzheimer's disease, we have been having these conversations for, as I said, over about a decade now, where we're trying to integrate biomarkers into both diagnosis of disease research and drug development. And so early on in that space, we had seen the ATN or amyloid tau neurodegeneration framework initially published in around 2011, and that had been very influential on our thinking about drug development, which was then informative of our Alzheimer's disease guidance that came out, I think it was 2018, we leveraged much of that information. And around that time, there was also research criteria published for the diagnosis, well, it wasn't diagnosis because it was defining Alzheimer's disease, because it was still a research framework, and staging. And so at that point, it was presented as research. There was still clinical diagnostic criteria that were used. Advice we were giving within the agency has been, and still is, that you can use clinical criteria, but you really need to use the research criteria based on biomarkers in order to make sure that you are accurately defining the population that will be enrolled in clinical trials. Now we're forward, in 2024, and those criteria, Alzheimer's disease criteria, have been presented no longer as research criteria, but as for clinical and research use, but they are spanning into clinical practice. We've also updated our Alzheimer's disease guidance, and it is aligned with that. And where this has come for us, the importance of this has come up with writing labeling and informing the design of clinical trials, is that we want to have a well-defined population. We want to then be able to describe that population that was enrolled in clinical trials in our patient labeling. So we have, in our labeling for any of the Alzheimer's disease drugs that we've recently approved, we have in section 14, which is where we describe the clinical trials. We do use the staging criteria in that section of the label. We describe stage 3 and stage 4 of Alzheimer's disease, but we also know that many prescribers still are not familiar with that terminology. So we have used the terminology as well: mild cognitive impairment and mild dementia. Now, we're kind of splitting both worlds. I really feel like we need to bring the worlds together. We really need to be able to say, this is the population we enrolled in the trial, this is the population we're indicating and proposing for treatment in the labeling, and we shouldn't have to be switching language from research to clinical practice. It's, I think, unnecessarily complicated for us, for patients, for prescribers. And so having these sort of diagnostic and staging criteria early, having that framework in place allows, I mean, it's a conceptual framework. We will fill it in as we go. We may not have all the pieces right at the beginning, but if we have that in place, it will translate over into clinical practice once we have the drugs that are approved and that we can use in those populations. So I do see this as having the diagnostic and staging framework as critical, bridging both research and clinical practice, because what is research now will become clinical practice in the future, and we need to be prepared for that.
Outstanding, thank you. And
Yeah.
clearly, CPATH plays such a key role in filling in the data to address those tools and gaps. I'm going to go back to you, Dr. Balabanov, and have you say a few words around Critical Path Institute's CPATH neuroscience programs with the EMA in paving path for pre-dementia early intervention in early days, and maybe you can say a few words around the new focus with EMA on including patients and the Horizons Alzheimer's efforts.
Right. So yes, there are certain ways to approach the EMA. CPATH have been very, very good in using those, as mentioned by my colleague Dr. Buracchio, to make sure that we do not deviate, as a regulatory world, does not deviate too much from the clinical reality. And our focus, of course, is that we are producing outcomes that are intelligible and usable by clinicians and patients. Now, what you mentioned, maybe I should talk about the fact that we have a number of platforms that allow us to engage with stakeholders and to make sure that we are fully aware of their positions, but also fully aware of their needs. And these platforms involve both early dialogue platforms in the sense of innovation task force dialogues that we can discuss with any stakeholder concrete innovative approaches and flag specific potential solutions or ways that these can be developed. It can progress into a dialogue with the agency where again, the CPATH has been very prolific and use this opportunity where we validate or discuss the potential validation of novel methodologies. And similar to what the FDA does, we can also issue some support letters and provide our positive support to certain developments in the field. And of course, we provide then more specific scientific advice for further levels of development, depending on the purpose and on the final context of use of the product. Now, in terms of engaging with patients, we are trying to have this at all stages, not only in the pre-approval stage, but also during the approval and in the post-approval stage. And we try to always make sure that the patient voice and the patient needs are covered. And as mentioned by Dr. Buracchio, that we do not have a situation where what is produced by research and what is evaluated by regulators is misunderstood and not practically used by patients and clinicians. So the way to interact with patients depends, of course, on the context, but apart from having dedicated working parties and specific specialists which are involved in our patient-centered initiatives, we at any stage can include patients in our assessments in both the pre-authorization and the authorization stage in the discussion of the benefits and risks of certain not only drugs but approaches. And as I said, all in all, this is an attempt from us at DMA to be always available to have a dialogue and to have an early interaction in order to guarantee that the stakeholders and the regulators do not drive a division between the newly developed strategies and trends. And we are always, as well as regulators, well informed and prepared when the time comes to be able to evaluate the data and the proposals that come to us. Have I answered at least partially your question, or did you have a more specific?
That's perfect. Thank you so much. I know that very few people are aware of the innovation that you're leading with patients and so much of what we do with you at CPATH. So I want to come back to our industry leads.
Am I allowed to go off script and make a quick
Please,
comment?
yes, you can go off script.
I just wanted, so I wanted to add that from an industry perspective, for those of us who are, trying to develop drugs and run clinical trials, the creation of guidance documents like the FDA and EMA have created, for instance, in Alzheimer's disease, has been incredibly helpful in at least two ways. First, and Dr. Baracchio, you sort of alluded to this, creating a common vocabulary that all of us across the field can use to describe different stages of Alzheimer's disease, for instance. The second way that it's been really useful for us is by clarifying where the goalposts are, what the objective is for treatment at different stages of the illness. Before people have symptoms, when those symptoms are incredibly subtle, and when they're less subtle. And so it allows us to try to develop endpoints and to clarify kind of at each stage of the disease, what is it we're trying to solve for exactly. A third way in which the guidance documents have been really helpful is that they're not overly prescriptive. They're future-proofed by being conceptual in nature and by describing what it is we're trying to achieve rather than getting so specific as to define individual instruments for measuring patients. So it's been really helpful, I think, to the field in general for this collaboration with health authorities.
Oh, thank you so much for those helpful comments. It's been interesting to see how these regulatory frameworks can really lead change to happen faster. I'm going to call on you next, again, Dr. Hirsch, because I think when we met, it was at the launch of the Huntington's Consortium, Critical Path Institute's Huntington's, and this is your background. Can you say a few words around your views of how biologic staging has impacted in Huntington's, and what are those learnings in rare neurodegenerative diseases that can help us in Alzheimer's and Parkinson's?
So for Huntington's disease, I think the biological basis has always infused it, in part because it's a genetic disease, and in a way, having a positive genetic test is sort of the ultimate biomarker. And as well, there's long been knowledge that the neurodegeneration in Huntington's disease is really profound prior to symptoms. And for example, in the basal ganglia, which is one of the areas that's very affected, about 2/3 of it is atrophied by the time of first symptoms. And so at the time of first symptoms, it's late already in the game for trying to treat it. And so that's an area where early treatment, pre-manifest treatment has always been on our minds. And actually, I've worked on a trial that was a preventive trial many years ago in Huntington's disease. So I think those things have been very important. I think the recent efforts to develop the unified staging system have done a very good job of using data that's been accumulated over the years, and a lot of that has been in really important natural history studies as well as in clinical trials, and a lot of that data has been brought together through efforts like through CPATH. And the framework that was recently published is very interesting. It's very data-driven, but maybe a little bit harder to translate clinically, because there are many nuances in it around the nature of the mutation, whether in ways that are hard to use clinically, perhaps. Another aspect of the Huntington's staging system, I think, is this concept of having a stage 0, where you have the disease from birth. That's also has the complication of at least when considered clinically, how does that kind of infers that people are tested And in Huntington's disease, at least in the United States and also in many places, most people don't want to get tested or don't have a reason to get tested. And so that kind of staging system in some ways isn't inclusive of all that majority of people who are at risk for Huntington's disease, but maybe they're at 50% risk or 25% risk. And bringing those people in in this kind of staging system. Again, the staging system is meant for research purposes, but at some point those gaps need to be bridged. And I think that's also a potential issue where genetics are used in these staging systems. But these are great, at least from my perspective, ways to start talking about those things and talking about all of the downstream implications of genetic testing and genetic privacy and medical privacy, which may not only relate to genetics, but also, for example, in Alzheimer's disease, where testing for factors like ApoE might say something about future risks of Alzheimer's disease, as well as future risks of potentially harmful side effect of this class of treatment. So there's a lot of complications. There's a lot of complications when you think about how these apply clinically and how to bridge the research uses of these frameworks with conversations in clinic and with that will be just amazing to see how they unfold out over time.
Thank you so much. I want to hear from Dr. Merrick a little bit about a transformational study he's leading with others to pioneer this path to prevention study that's intersecting the worlds of natural history study and clinical trials. Can you tell us about that and what led to this?
Sure. So maybe even before that, I just want something came up as everyone was talking. I'll just mention it. One of the, I think, really happy circumstances that's occurred as the biologic definition of these disorders becomes more evident is that there has been collaboration among the disorders as well, so that obviously the NST framework has learned a lot from the Alzheimer efforts and from the Huntington's efforts, and I think, of course, with regard to Alzheimer's disease and Parkinson's disease, there is shared biology, and I think so really enabling us to maybe transform our thinking one step further and think about how can we really understand the biology of neurodegeneration as opposed to simply looking at these as more siloed efforts. And I think that's a future that is probably coming at us fast and furious. And as we, one of the things that we always think about, at least I always think about, is that we're all neurologists on this podium here. Neurologists are always about 20 years behind cardiology or oncology, and I think that here again, I think as we think in the future, you can envision a way in which, based on the biology, we can begin to bring this into the clinic, testing individuals at an early stage, getting your neurodegenerative battery, just as you get an oncology battery or a cardiac battery in the future. I think that's where we're heading, and I think that's something that I think is sort of not here today, of course, but is sort of within our grasp. And now getting to your question, I mean, it's in the same way, trying to understand, as we've all discussed, it's clear that these neurodegenerative disorders begin way before symptoms arise. There's no question about this. So the time to treat them is before symptoms arise. And so that's, of course, challenging in many respects. One of the things we're trying to learn from in the PPMI study, having enrolled several hundred, several thousand individuals now who are prior to the onset of symptoms but have biomarker evidence of what we would call neuronal synuclein disease. Can we begin therapies at that time? This is something that's already been brought forward in Alzheimer's disease, but we're bringing this forward in neuronal synuclein disease based on a definition of their stage of disease. So we hope this is meant to be a learning study at present, but we hope this will rapidly provide the learning that will enable this to become an actual study that leads to pivotal studies and new therapies at that time prior to the onset of symptoms in what we are calling stage 2. Again, not prodromal, but stage 2, to try to be clear about what we are, who we would like to treat.
Incredible. So I think for the last commentary from each of you, what I'd like to do is to turn to each of you and recognize that we're here in the room with people representing a whole variety of different diseases, not just nervous system disorders. And if you were to speak to someone that said, my disease is just so complicated, this is too difficult, staging is never going to happen, it's centuries away, what would you say to them about when is the right time to consider biologic staging? So I'm going to start with you, Dr. Balabanov. We've heard from you and Spiros around come to the EMA early. So this is kind of putting you on the spot. But what would you say to somebody in the new disease areas that wanted to contemplate this question?
Well, of course, like everything in life, we should start with small steps and look for small successes. And I do not like the expression "go for the low-hanging fruit" for many reasons, but sometimes I think we're seeing this in, let's say, in my 15 years, more than I would like to. I have seen this approach where researchers, both patients and academics and clinicians, are aiming for the golden grail, the big solution, and it seems often, as you say, either too far out of reach or completely impossible to reach. And they come to us and say, this is an extremely impossible situation. There's nothing to be done. And in that sense, often small achievable goals are being either not pursued at all or being completely omitted. And from the experience that I have, but also with my colleagues, it is due to incremental successes and things that generally are perceived initially as small successes that we, as a field, together learn and progress towards the final ultimate goal of solving the problem in those diseases. So to anyone who has this perception of, oh, this is too difficult, it's impossible to achieve, I can give a very practical example of remember what we did with multiple sclerosis 30 years ago. And remember the first attempts at curing a seemingly unsurmountable problem, and the initial perception of how little certain achievements were considered to bring to the patients, and see where we are now in this disease. And I can only say that due to the initial push, the initial small successes, the whole society of stakeholders were actually successful in the end in developing the armamentarium that we had now. So if we're only aiming for the seemingly impossible to reach results, we will miss the initial small steps that are extremely important. Now, when to come to us, there's no early enough stage. As regulators, we'll be ready to speak to anyone as early as possible and to give this message. So I would say the moment that there are ideas, that there are proposals for certain staging of a certain condition, come and discuss with the agency, and the agency will have at least a position to guide the further research. And of course, as mentioned before, we will not be ready immediately to write a guideline, but we'll be there with the key opinion leaders on our side to tell you where and which type of efforts we'll be ready to support, so the successes can come and can probably lead to a much more coveted result. I stop here and let the others speak. Thank you.
Excellent, and data tells the truth. So I'm going to go to you, Dr. Hirsch, and say some words, what you would say to someone in a new disease area.
Well, yeah, I mean, I think I would certainly echo what we've heard before, the importance of taking small steps, the importance of working with regulators, the importance of having some of that work not just be between a given company and the regulators, but in fora that are more representative, such as CPATH represents. All that lays a lot of groundwork for moving the way regulators can think about development and the way the field can think about development. So I would certainly echo all of that. I think also there's clearly a role for technological innovations, like the development of the synuclein seeding assay, for example, which that development will drive lots of change in the field of synucleinopathies, and it will stimulate even the development of additional biomarkers that will further fertilize the field. As we've seen in Alzheimer's disease, where the pace of biomarker development and their potential applicability for research and for clinical use has been just mind-bogglingly fast, and will profoundly affect how neurologists practice.
Excellent, thank you, Steve. Jeff?
My message would be, don't let perfection be the enemy of the good. We have to start, I think the right time to start with biological staging is when you have the first tools available to start to interrogate the biology in vivo. Look, it's going to be iterative, and we're going to discover over time that some of the biological stages that we thought were important ended up being too granular, and we're going to discover that other aspects of the biological staging is too coarse. But we have to, I think we can all be aligned at what the end goal is. The end goal is that we create a framework of understanding the disease and matching the right therapies to the right patients at the right stage of their illness. And nirvana in the future is where that biological staging scheme perfectly matches the tools that patients and the doctors caring for them have to intervene in the process.
Excellent, thank you. Ken, I think we're living this today in the LSD world.
Again, of course, I agree with what's been said so far, that I think one needs to really begin to integrate biology at a very early stage. There's no, in some sense, it's obvious that biology is important in disease. It's crazy not to think that it isn't. Of course, it may be challenging to understand fully what's the complete biologic underpinning of a disorder, but I think starting with what you've got and moving forward in an iterative way makes perfect sense. I'm going to just raise one other point, is that I think it's important as we continue this effort that we also try to make these biologic tools that we are developing accessible to widely. So far, the technology is great, and of course we will start with highly technical tools, but I think we also want to be very agile about trying to make these tools rapidly more accessible around the world so that they're available and we can understand disease more broadly. That's another big challenge for us, but I think that we shouldn't be cowed by the difficulties. I think this is the way we're going to make progress. Yeah.
Excellent. And so I'll call on Dr. Baracchio. This is a topic I know you've brought to our attention at
Yes.
CPAP.
I don't think there is such a thing as too early to start. I don't even think you need the technology yet. I think we're talking about a conceptual framework. This is a conceptual framework. You can start thinking, even with a clinical mindset, as to what the stages might look like. You will develop the knowledge. You will fill in that framework. It will be an iterative process. It will be you will adapt it as you develop new technologies, as the science evolves, as our knowledge evolves. That's the important thing, is that these are not static frameworks. I'm sure, although we've got right now the Alzheimer's disease diagnostic and staging criteria tied to amyloid largely, and NSD is tied to alpha-synuclein, I'm sure if the frameworks that are in place, despite the names and despite those biomarkers, if we came out later and said, you know what, alpha-synuclein is not the important thing, it's something else, you know what, you can tweak that framework.
Exactly.
It's there in place and it can change, and the same thing with amyloid. They're there, but they can adapt. And so I think it is important to come in with the end in mind, play the long game. What the criticisms I've been hearing about the current staging criteria that have been proposed have been tied up in the details. We don't know enough. We don't have that information yet. How can we make these bold statements when we don't know that amyloid is the cause of Alzheimer's disease? It kind of doesn't matter. We will update it as we go. So I think it's important, if we get that framework in place, that will help us later. As I said, current research becomes future clinical practice. If we have that in place right now at the research stage, it will inform how we practice clinically later.
Outstanding. I think you could tell the enthusiasm for this session really centers around Critical Path Institute's excellence in model-informed drug development, patient-focused drug development, everything data. What an incredible session. So thanks to the distinguished panelists, and we look forward to continuing this discussion throughout the next days.